Got it.
Okay, let's get this party started. Welcome to our GRG meeting. Today's date is August 28, 2021. Special thanks to Miles Jacobs, who handles the mechanics of the emails and setting agendas and keeping all this on track. Thank you, Miles. You're just doing a great job, and everyone here really appreciates it. We have some great scientists and great members of GRG. I think I know all of you personally, and each and every one of you are doing things, making contributions to advance our mission of aging intervention and biological age reversal, even ultimately someday. So really glad you could join us. Our topics, we can talk about anything during the open discussion at the end, but on the agenda, I see that Miles put together and we collaborated on. He says the 7-day reset of the embryo epigenome. I have to change my glasses, hang on. To age 0. Well, as reported recently in the literature. So, Miles, it sounds like you know all about that. Do you have some information to report on that?
Hi, Johnny. Hi, everybody. Yes, it was quite a surprise to me. I'm going to throw the paper up on the screen, if you don't mind.
Yeah.
Let me just do that. Hang on a second.
Yeah, there was a conference on ending age-related diseases last week, and I don't remember his name, but the director of the Buck Institute gave a talk in which he included that particular result as part of his research. Very curious that the embryo resets itself after 7 days rather than at the very beginning, but it seems to be the case.
Very exciting paper, for no doubt.
There was some speculation of why that is, but nobody had any very good ideas.
Till now, we used to think that germline cells don't age, but apparently they do, and the reset happens actually during embryogenesis. So that was a revelation.
Mm-hmm.
Yeah. And Miles, are you working?
No, I'm just struggling. Just struggling now. I don't understand why it's not sharing. Let me reset the screen and see if it now sees it.
So you're pressing the share
right,
screen.
Here we go. There we go. You can see the rejuvenation effect, amazingly, appears to be on day number 7, the 7th day. On the 7th day, the embryo, both the sperm and the egg are actually aged. They're not epigenome age 0. They have epigenetic damage. They have methylation damage. But on the 7th day, after its formation, the embryo resets itself to age 0, which is most interesting. I never expected that. I don't think anybody else expected that. We all thought, I think it happens at conception, but no, it happens after about 7 days.
I have a question. How many cells are in the embryo at that stage?
Sorry, Paul, I haven't even thought of that. I don't know if anybody else can answer.
It was Gladyshev who gave the talk I was mentioning, and he showed a picture, sort of a cartoon, and judging from the picture, it looked like there were around 100 cells in a sort of a ball at the time when that happens. But maybe I'm not remembering right.
I wonder if that is the point of time where occasionally an embryo will split, and there will be identical twins, as opposed to 2 eggs coming and being fertilized.
Mm-hmm.
Well, the interesting thing is it's not just one cell. It seems to be the old embryo, which is that has implications for anti-aging.
Well, this could be another time when it could happen, I would suggest, since every cell now is back to age 0.
Yeah, if you have a ball that's made out of cells, it's conceivable it might fission into 2 balls instead of 1.
Yes, that's my thought.
So yeah.
Or 3 or 4.
Right.
Well, or 20 trillion,
Yes, right.
which is what we are. We're about 20 to 30 trillion cells. So this happens after the embryo has started creating itself, and it's long after conception. I found this most interesting.
Totally agree. I would rank it as one of the best papers for this year.
Well, it's very interesting, but I don't know how it applies to anything that relates to aging at this time. It isn't obvious at all that it would apply to anything that we would be interested in from the point of view of our futures.
You don't want to have your cells reset to 0.
Yeah, that relates to a question I was about to ask, is how are we going to take this information and related information to turn it into a therapy that we can use, like sooner rather than later?
Yes, that is always the question about all of these research results, right?
Right. So I'm on the case. Resetting the epigenome to a more youthful state in us is one of the things that it's one of the top 2 things, actually. I think recent information that has pointed to how difficult, how challenging, we're not going to let this put us off, but how challenging gene editing and gene therapies will be. But now, my new number one is resetting the epigenome. And I think part of that is a good first step. And I'm going to look to this group discussion and some brainstorming here, is to identify scientists who are working on this right now. I guess the scientists who wrote this paper, hopefully they're accessible and approachable. But others on my list along the same topic are Juan Carlos Belmonte at Scripps, and also he has a number of less senior but very talented scientists working in his lab. I've collected their names, and also Rodolfo Goya. I don't know Rodolfo personally, but I know some people who do.
I know him personally, and I can introduce you, Johnny.
Oh,
Goya
thank you.
is a fellow author in one of our papers, so I can do that.
Outstanding. Also, I have in my notes, there's a Spanish group led by Jesus Avila, who has close ties with Belmonte. So, go Spain. I guess Belmonte is originally from Spain. And so who else do you know who's working on this?
Well, there's Turn Biotechnology, which is a spinoff of the Stanford group that did some of the early work on this.
Yes.
They combined the Yamanaka factors with a couple of other enzymes that are present in embryos, and showed that that when you stop the process in like 3 days, that you can reset the cells to a younger epigenetic profile without having them lose their programming about what kind of cell they are. There was a talk at this conference I was referring to by a guy from Turn Biotechnology, and there was another guy who was working in the same area, and they seemed to be competitors who didn't want to recognize each other's work, if I got the vibes right. But they were both saying more or less the same thing, that you can rejuvenate cells to a certain degree by using some subset or superset of the Yamanaka factors, but you have to be very careful to stop the process after a few days rather than doing it continuously, and that different cell types require different rejuvenation times, which is going to make it quite complicated because you can't just do it to everything at the same time. But as I understand the Turn Bio idea is they want to do something like the coronavirus vaccine, where they use RNA, messenger RNA that will express the enzymes they want to implement, and they put them in a liposome and inject them and hope that the cells that they're interested in are rejuvenated. It's not clear exactly how well that works yet, because they've done it on rats with some success, but nothing, and they've done it on human cell cultures, but nothing beyond that. But my feeling is that before you rejuvenate cells, you should clear out all the senescent cells so that they don't, because they have a certain potential of becoming cancerous if you start messing with the Yamanaka factors with a senescent cell. And so I think the first step is you sweep away all the garbage, and then you try to rejuvenate what's left.
Yeah, thanks for that reminder.
I'm an investor in Turn Biotechnology, so I have a somewhat vested interest in their success, but they're moving along, but it's a little hard to see exactly where they are at the moment. One of their ideas was to simply take fat cells that you got by something on liposuction from a subject and apply the same technique continuously until they become pluripotent stem cells, and then re-inject them in places where stem cells are needed. And I think that's something likely to do a long time before they ever achieve anything that's actual rejuvenation.
Yeah, well, thanks for the reminder, John, of the importance of Turn Bio and other companies in accelerating all this and harnessing the forces of free enterprise. And
Right.
it's understood that to do their good work, there's a different model than a university or academic research model, and all sorts of regulatory and financial accountability.
Oh yes.
And
the FDA is blocking the way very vigorously, I'm pretty sure.
Oh, I'm sorry, someone's blocking the way?
The Food and Drug Administration.
Yeah, well,
it's going to be a long time before you can do anything like this to humans because of FDA rules.
Well,
even if it's spectacularly successful.
Yeah.
Well, that's actually easily bypassed. You can just go elsewhere in the world and do it. I don't think the FDA is blocking it.
Right.
They can block it in the US, but it's not. I think it's really the science. We
I'm
have
sorry to be
too much
provincial. Yes, it's
expectation
true,
for a science which is very basic.
Yeah.
Yes.
Well, also, even here in the US, a certain amount can be done under the under an IRB approved clinical trial. And I think Dr. Zalzala joined us today. He has a telehealth company, and something we could touch on later is that I'm bringing about a telehealth concept called Age Management Telehealth Physicians, where I'm managing the thing, and we're in the very early stages now, and it will be under not only it'll be under an IRB umbrella. So, but also we've had discussions in a group of business people and scientists I'm involved with to set up offshore clinics in the Bahamas that would be easily accessible. And there's that, but all this is in the future. I have heard that these companies they have to have a disease condition to get FDA approval, to get money, to get investor interest, that sort of thing.
And aging is not a disease.
Pardon me?
And aging is not a disease, according to the FDA.
Well, right. But still, some of them I'm hearing, and everyone in the group here who's had more experience than me, tell me what you think. But they also, they're keenly interested in solving aging and in their own biological aging, which is pretty tremendous motivators. So they might run a a shadow trial where, okay, they have biomarkers and objective measures that are up on the clinical trial documents that you'd read about on clinicaltrials.gov, but also run some other markers that are more closely related to biological aging. And of course, DNA methylation comes to mind as the top one there. And
Yeah, that's
number
actually
of
what
others.
that's actually what our goals are for the rapamycin trial we're doing.
Yeah,
Is our primary outcome is visceral fat by DEXA scan, but then kind of our secondary kind of looking at as many biomarkers we can for aging. So something like that would be great. I mean, if you know when and if we finish the rapamycin trial, we hope to move on to other things. But I tell you, clinical trials are not easy to get off the ground. But
yeah.
and not impossible, just not easy.
Yeah, I don't want to derail us with a sidebar, but I've had some interesting personal experience with rapamycin recently. Maybe in the open discussion later, we can talk about that. I'd be interested in hearing what you can talk about anyway regarding variability of its effects in your patients. But getting back to well, what other companies aside from Turn Bio are working on resetting the epigenome to a more youthful state?
Alkahest is, you were just mentioning about companies needing to focus on chronic condition or a disease. Alkahest has been doing that, and they are probably ahead of all other aging research companies. They have already got 3 products in phase 2 with FDA. So, we do get worried that FDA and the pharma lobby will block. But in the case of Alkahest, there has been, no such deviation or delay so far. They've been moving ahead quietly, but very successfully. Probably next year, we might see phase 3, and then maybe the following year, we might have first anti-aging products in the market.
Oh, really? Specifically for anti-aging or biological age management?
They are going after individual, chronic conditions, like you mentioned, Johnny. So one of them is Alzheimer's. They've always been focusing on that, which is kind of really tough target to go after. I wish they had gone after a more easier one, like type 2 diabetes, or where the reversal could have been much more pronounced. But they have gone after neurodegenerative diseases. So that is one of their applications. Or I think 2 out of the 3 are neurodegenerative.
What technique are they using?
Sorry?
I said, what technique are they using? What's the
They are using similar to what we are doing, which is they are extracting fractions out of plasma.
Oh.
And they were very fortunate. They got good funding very early from a European company that deals in plasma called Grifols. So they also got a supply of a lot of kind of a young plasma that they needed, and also a lot of money. Now Grifols has bought out Alkahest completely. So earlier they had invested $50 million or so, but last year Grifols, I think, bought them out for $340 million or something like that.
The question I always have for people who are doing plasma things is, okay, if you give them a treatment with your plasma extract and it produces beneficial results, do these beneficial results last, or do they fade away after a few months?
No, it's a very good question. And, our own experience has been that it fades away. So there would have to be repeated treatment for life if you want to continue to enjoy the benefit. But Alkahest has a strategy where, yeah, I agree. Alkahest has but something is better than nothing, and especially if people do have some chronic condition that it's helping with. But in case of Alkahest, what they have been doing is a little different. They have identified what are the proteins in plasma that go up with aging and go down. And then they've identified which are the good ones and bad ones, and then according to them. And then they have come out with certain, techniques to block the bad ones. That is the strategy. So they're going after that, and then through that they hope to address or at least mitigate some of the neurodegenerative disease.
There was a long discussion in the lifespan event. We had a rejuvenation webinar, and one of the nice comments was, I think it's Professor Dr. Grissom, was that the way of clearing the blood like this is not going to be the be all and end all. This is just to set the table, because you're going to still add all the interventions later on, but you have to clear the blood first. It's almost like, when you're planting wheat, you first have to plow before you actually put the seeds in. So, and it's not surprising that the effects wear off, because the body is constantly aging. So, but you can't avoid doing this, even though it is going to disappear after a while. You have to do it, and you have to keep on doing it. It's the same thing like plowing a field. Otherwise, you have to deal with weeds all the time. You can see these aging molecules almost like weeds that will upset your wheat field. So the pathogenesis, or what I forget now the name of this, parabiosis that you do in the beginning is always going to be necessary. Like any experiment is almost like washing your lab equipment. And then you start with your Yamanaka factors, and then you can do whatever the turn bio treatments will be. You can do your gene therapy, but fundamentally you've got to first clean up before you do the rest of it.
Yeah, I think that's what I was saying earlier, that you want to clear the senescent cells before you start these other things. But Harold Katcher and his elixir claims to have reset the epigenetic clock by a factor of 2, and that in principle is a permanent effect, not something that you have to reset every couple of months. So it seems that there are things in young plasma that can actually achieve some kind of permanent reset, although those guys are very reticent to tell us what exactly they're doing and how it works. So I would like to have as much information about it as possible, but nobody in Katcher's group seems to be talking.
Well,
John,
Akshay is with us today. Akshay is there you go.
Yeah.
You take it, Akshay.
Yeah, so John, I am a colleague of Harold.
Okay, I didn't recognize your name. Okay, so tell us how things are.
Yeah, no.
Have you done anything with dogs yet?
Yes, no, we have a lifespan study that's going on. That was sponsored by the Heals Foundation. I must mention Didier. I don't know if anybody knows him, but he's been a generous sponsor, and he is open to sponsoring other rejuvenative therapies. So if anyone is looking, Johnny, then you could point them to Didier. The lifespan study, now we had started with 8 female rats.
Lifetime? You're talking human lifetime or animals?
No, rats.
Rats, okay.
Yeah, so lifespan. Human lifespan would take very long. It should be done, but you won't know the result till 30 years or maybe more. But with the rats, we have 8 female rats and treated, and 8 are untreated. They started the trial at 24 months. They are now almost 30 months. So we have done 3 dosings. So the point that you mentioned earlier, I would say that there is an effect that wears off, like Johnny said, or someone mentioned here. So the effect does come where there is a reprogramming, but then the aging program continues and it does wear off. So there is repeated dosing that's required even with Harold's protocol. And we have done 2 dosings in the last paper that we had with Steve Horvath, where he did the, BioAge clock, testing. But in this one, we have crossed and made a 3rd dosing The results of that are expected on 2nd September. If anyone is interested here, I can share it with them.
I'm
And
certainly interested.
yeah, we will, then I will share with Johnny, and then he can share it with anyone who's interested. And so, yeah, so we are presenting to the Heals Foundation on 2nd September. We are hoping that there will be an improvement from the second dose. If it is, then that's great. Then we are moving towards the, in the right direction. Yeah,
Harold
so
was talking about
lifespan.
Excuse me, Harold was talking about applying the substance to dogs, and I was wondering if anybody's.
Yes, that's next. That's next. So we have set up a lab in California, and right now Harold is busy developing enough E5 that we call, as we call now, elixir, to inject into dogs. The colony is available, and we're starting with one of our friends, his dog Haka, who's in the last year of their life. So that's the first one we will test, and then we will start the study. But going back to the lifespan, yeah. So the known lifespan of the Sprague-Dawley rats that we've used is 36 months, and they are at 30 months. So in the next 6 months or 12 months, we will see a lot of deaths, and we are hoping that the treated last longer or live longer than the untreated. And if that happens, that'll be great news for all of us, because finally we have some lifespan extension that we can show.
Very good. What's the average lifetime of wild rats?
Wild rats, so it depends on their species. Each species has a different kind of. We on this one we are working with Sprague-Dawley, and Sprague-Dawley they are 36 months.
Mm-hmm. Okay.
That's their average lifespan, and most of them naturally die of cancers. Most of them. So it will be interesting to see whether E5 has any effect on cancer. We have hired one medical director now who is ex-faculty from Harvard University, and he's a practicing doctor. And he wants us to do a mice study with cancers, before we can inject this into humans under IRB. Like Johnny said, we hope that we get this IRB permission next year to try it on humans. Harold himself is very keen to try it. Yeah. So we might be doing my study on cancers to see if there is whether it it flames the cancer, or whether it subdues the cancer, or whether there's no difference. We want to find out, obviously. So all these kind of parallel studies are continuing. These things take a lot of time, but yeah, there'll be a toxicity study as well, and we need to complete all of that before we can go to FDA and file an IND, just like Alka has. So, but we are in that process. We are also going to tie up with a university like Stanford to do a lot of characterization work, standardization work, which is required before an IND application. So all the anti-aging research companies have to realize that we have to follow the path of the pharma companies, whether we like it or not, in trying to create very standardized doses and things like that, rather than supplement companies, if we want this to come out as a prescription drug available to everyone. So we are also following in the footsteps of Alkahest. I think they've set a good example.
Well, I'm still here to help, Akshay. Call on me anytime.
Yes,
whatever
you've always been
resources
very helpful. Thank you, Johnny. Thank you. You've always been very helpful.
I would ask a question, Akshay. Thank you very much for that. That is a fantastic update. Really appreciate that. Very exciting. Just a question, I presume, in the case of this, or not a question, a comment, in the case of this 7th day reset, I'm hoping to see that people then try to reset on the 14th day, see what happens. Reset on the 21st day, see what happens. If it's possible to reset continuously, taking baby steps at a time. These are mouse and rat embryos. I believe that whatever is going to happen here must be applicable to all mammals, because I mean the fundamental process, and I think this one I'm going to ask Akshay about this. The fundamental process appears to be that the differentiation is through the effects on the Hox complex of the repressors, which is the polycomb, the polycomb repressors, which ultimately is seems to be under the control of the terra RNAs. So it appears that as the embryo splits its cells, it applies repressors via the polycomb repressors onto the Hox complex or Hox cluster of genes. And as it applies different sets of repressors, that determines the identity of the particular cell line. So I'm just curious for Akshay, have you people been thinking about this? I know the Liu group, they've been thinking very carefully about exactly how differentiation happens, because it appears that the OSK factors, the Yamanaka factors, are affecting things like the polycomb repressors and the Hox complex.
Yeah, so our research is in a very different direction or using different mechanisms. We are following the work of the convoys, which is where they found that there are factors in the blood, young blood, which influence when circulated, they influence the aging. But with regards to what a lot of companies, Johnny was asking the name, even George Church and some of his group companies are doing things in related to cellular reprogramming and Yamanaka factors, partial reprogramming. Vadim Gladyshev, I think I really admire his work, and that's why I love this paper. I mean, this is definitely a great revelation. All of us used to think, as I said, that germline cells don't age, and he's put a spotlight on something that is extraordinary that happens. I mean, this is why probably we die. I mean, nature is created this recycling so that all the errors are wiped clean, and a brand new specimen is born for survival of the species. This mechanism that you're mentioning, it's not so complex. It can be studied, and I'm sure there will be, maybe in Gladishev's group or other groups, they will definitely try to, do some studies based on that mechanism. Repressors using RNA. I mean, today RNA technology is moving very rapidly. We have seen that in vaccines, mRNA was used, and now Turn Bio, as John mentioned, has licensed an mRNA technology from Stanford, and they are planning to use it as well. So being able to make such changes in the cells looks like science fiction right now, but can become reality in the next decade. The only issue so far with whatever studies that have happened with partial reprogramming of cells has been that stability. The stability after the programming is done has not yet been completely solved. So it does lead to genomic instability in some cases, which is uncontrolled, and it does lead to cancers. That doesn't mean it cannot be solved, because all the mechanisms that nature uses, we can create biosimilar mechanisms. So it definitely has a lot of potential. The difference between, let's say, our therapy and what one can do with the reprogramming is that something done through the factors circulating in the blood, they have a life. And therefore, once they are exhausted or they do the function that they're supposed to do, one needs to again, flood them into the system to continue to maintain that benefit. So they, unfortunately, they would need to be, there would need to be dosing for all your life. I mean, on a regular basis, we have to find out what is the interval between doses. Harold thinks it could be 1 year or 2 years, which would be good. I personally suspect it could be 90 days. But so that's the challenge with it. But it is much safer than cellular reprogramming because there is no scope for any kind of instability as it's using actors manufactured by nature. And there is no human intervention. But the advantage with cellular reprogramming is that you could do permanent resets. Like what has happened here in embryogenesis is permanent. I mean, the errors, it's a clean slate that's coming and then everything is going from there. So that's the advantage. So you might be able to get away with one treatment and then maintain your youth. So that's my take on it.
And I'm after John, but John, you've been enthusiastic for a moment.
Yeah, at this conference that I listened to last weekend, the Ending Age-Related Diseases Conference 2021, there was a guy who gave a talk in which he was doing something very similar to the Turn Bio thing, where he was focusing on individual cells and looking at what the Yamanaka factors do to them. And he addressed the question of the reset without forgetting your programming to what kind of cell it is. And his work seemed to imply that there was sort of a valley of stability, and that if you overshot the reset, that it would settle back into this valley, and the cell would retain its knowledge of its function. And I consider that very good news because it means that you don't have to hit a precise target. You can overshoot a little bit and still get rejuvenation without having the cell lose its identity as a liver cell or a kidney cell or a lung cell or whatever. And so that I thought was an important observation. Although how solid it is, I couldn't really say because all I heard was a talk. I haven't seen any publications about it.
Yeah.
It would be
Do you
very
recall, or
good
could you look up his name, John?
I was asking the same thing. Yeah, I'm curious because this is a new player and we would like to know who it is.
Were you thinking of the speaker from Calico?
Maybe it was, yeah.
Yeah, and he was using a different factor, MSX1, to rejuvenate myocytes. That's a transcription factor that's expressed when salamanders regrow their limbs.
Yeah, I should have prepared for this meeting by having the agenda of that conference in front of me, but I didn't. So I'll send an email to Johnny
But yeah,
in the group.
Calico, we know.
Saying who exactly this was.
Yeah, great. In a moment, I'll show you some data and a graph. But regarding the idea that was mentioned earlier about having the right types of proteins, and I would suspect that's different for different people, but how do we remove or decrease specific proteins associated with aging? Or I'll focus the question, how do we remove specific proteins? Or how is Calico or Alcas doing it?
They are using binding factors. They bind them and then degrade them before they can be functional. So they're using that and they file patents for that. But it's something very common in Western medicine. For example, we all take statins, and statin does a similar thing. They block certain enzymes, and so it's not something complex. It can be achieved safely.
I think antibodies specific to particular factors would be okay. That's what you're describing,
Yeah.
actually.
Yeah.
Yeah, that could be.
Yeah.
There's one protein called CD38, which apparently increases is produced among other places in the SASP of senescent cells and rises as you age. And apparently it eats NAD plus and takes it away. And so there was a talk at the meeting by a guy from the Buck Institute who was claiming that the reason NAD plus goes away as you age is because the CD38 is rising, and there's sort of a leak in the bucket. The NAD plus is supposed to be recycled, but this breaks the cycle and diverts it away. And that there was some evidence that most of the CD38 is coming from senescent fat cells, and if you could clear those, you might patch the hole in the bucket. Also, Apigenin, or if I'm saying it right anyway, the supplement apparently is a CD38 antagonist and removes it. So that automatically should boost your NAD plus without having to take these supplements that are supposed to provide the basis for making it. So that's one protein that one would like to get rid of. And although I guess it has other uses, but you wouldn't want to remove it completely. But that's one protein that there's apparently a known antagonist for.
Just to comment, the reason for death basically is because it's much you was based on half-life. So if you have a new organism, it starts with a clean slate. You've got a period where the proteins will hang together, they won't malfunction. And by the time they malfunction, biology has organized reproduction. So the old organism can be allowed to malfunction while the young ones who are now born again have a new half-life. So half-life follows half-life, and that's why the old organisms are allowed to die. And genetically, biochemically, that's much cheaper than trying to maintain the old organism. So biology has found a cheap way to keep life going. That's the problem. So that's fundamentally the problem. It's biology has found a cheap way to keep the species going by allowing the old to die and then getting a new form, which has a half-life basically for free, which will last until it can reproduce.
Except that there are organisms that are known that don't seem to have that sort of biological clock ticking that causes them to die of old age. So it's not universal. One thing I heard that I hadn't heard before at this conference, which is that this methylation business of methylation to epigenetic program is unique to vertebrates, and that invertebrates have different ways of expressing genes that have nothing to do with methylation. And so I thought was an interesting insight, because I would have thought it was a lot more universal than just vertebrates.
Well, are we ready to move on to the next topic?
Which is gene therapy.
I don't know. I have, like, personally, I have all day. We have such an all-star cast here. I want, I'd like for, to make this not too long for you. But okay, the next one, gene therapy advances.
Johnny, there's
I'm
a
hearing
question.
some pessimism.
Johnny, there's a question in the chat.
Go ahead.
There's somebody, Zahad, okay.
Oh yeah, Sajad.
What was that? Somebody asked, what was the supplement that they mentioned? And I didn't catch the name either. It went by quickly. It was something like epigenetic.
I'll type it into the comments. I was in the middle of doing that.
Yeah.
There. I'm not sure I spelled it right, but I think that's it.
Epigenin, okay. But certainly a search would find it. A Google search would find it with your description.
Yeah. Interesting.
It's in many LEF supplements, by the way, and I've been taking lots of it for years.
Yeah, senolytic activator. And that is one of the supplements that Sandra Kaufman recommends as a part of her protocol, which I, for what it's worth, I personally follow that in my own age management program.
Yeah, it's sort of known to have something to do with NAD plus, but it wasn't the exact mechanism apparently hasn't been clear until recently. And so that's the new information.
Yeah. Okay, gene therapy advances, gene editing, possibly knock-ins of more youth-enhancing genes. Anyone have anything to talk about there?
Johnny, I just wanted to share the information about CRISP-off, which is it's specifically epigenome editing,
Really?
which I saw come. It's very specifically epigenome editing. So I've got a paper. I'm just going to throw it up, but that's my my comment for this at this stage.
Did you? It was hard to hear. Did you say CRISP-off? How do I spell that, or put it in the chat, please?
Okay, I'll do so.
Oh, there you are.
Now. Yeah, it's good. It's on the screen, Johnny. It's on the screen.
I see it. Yeah, thank you.
So very specifically, epigenome editing.
And how far along is this? Or I should say, how close is it to being practical and something we can use to stay younger?
To be honest, I have not read much more. It was interesting enough as it is because it's specifically targeted to epigenome editing and not to normal genetic editing. But
Yeah.
it seems to be, the paper seems to be quite efficient. I'm not sure. There was talk now in the discussion about 20 base pairs, errors and so forth. That was also new to me, but I just found this notable that this is CRISPR specifically for epigenome editing.
Amazing. Okay, something I wanted to run by all of you, a concept that I've had, like when I think of a couple of luminaries, for example, in resetting the epigenome, the person who first comes to mind is Juan Carlos Belmonte. When I think of gene editing and gene therapy, the person who first comes to my mind anyway is George Church. And, those guys, they're involved in so many things. It's so hard to get their attention. I've thought in order to truly make some advances, it would be to approach those working in their labs who are working on the kind of projects that they know about. And, these people, let's face it, they may be anxious to be striking out on their own in their own labs, get out from the shadow of the, the big guy or the big gal, whoever's, heading the lab. And this, in a way, it assumes, it assumes like a lot of money to set them up in a lab or even just set up their own research. But I've, one who I've had in mind is to contact, I forget her name. It's in my, I have so many files open, but one of the researchers in Belmonte's lab, Paloma Redondo is her name. And just, ask her if she'd be willing in doing some things that we would specify, but, we would, again, assuming there's money there, we would provide the money and, the project overviews. But what do you think of that idea? Have any of you ever done that before? Does it seem like it would be fruitful? Okay, no ideas. You don't know till you
Johnny,
try.
no, Johnny. I think it would be very fruitful. In that ending aging conference, there was a talk by, I think it's Dr. Grisson. I'm not sure if I'm pronouncing his surname correctly, but he has two clinics, I think one in Colorado and the other one somewhere in New York, or on the East Coast. And he was saying that we can now start to do actual wellness clinics based upon anti-aging. We have the aging metrics, like your anti-aging metrics, which you've got online. We then can do the supplements based upon the results of the metrics or the biomarkers. Then once we've done that, we can go to the new techniques like the parabiosis experiments, and then also ultimately to gene therapy. So there is a pipeline you can see now for anti-aging. It wasn't the case in the past. And many of the elements of the pipeline are successful and well understood. The parabiosis is successful. It's getting very well understood. And the same thing with the supplements, like starting with metformin, adding vitamin B3, niacin, and so forth. So I think, I mean, he was saying that lots of people have been trying to get him to franchise his clinics. I actually think that it's, you know, in your case, GRG, we should collaborate with an actual wellness clinic and start to test these things. He's been doing it. What we really need is just a medical doctor. And I think there's lots of medical doctors in the GRG group. So I don't see why not.
Well, so are you talking about therapies that are available now, such as what I've come to call the usual suspects, senolytics, you know, rapamycin, GDF11, some blood-based therapies, umbilical cord plasma, the kinds of things the Comboys and Dobry Kiprov are doing, the therapeutic plasma, they call, I think it's generally called therapeutic plasma exchange. I think of it as therapeutic plasma replacement. Are you talking about testing the kind of things that are available now, or developing new therapies from current basic research?
I mean, I think.
Go ahead.
I think the question is, what is stopping you or GRG from collaborating with a doctor and doing that?
What is that?
From doing things like, for example, checking up on the results of metformin, checking up on the results of niacin, then having a go at the parabiosis experiments, even if it's just with mice. But I suppose we could start slowly with humans, because Russians have been doing this. I'm not saying you can do, you know, the whole hog, but there are clear interventions like the biomarkers is a very good beginning, which you're already doing. You're already doing the biomarkers, which predict the kind of interventions with metformin or rapamycin or whatever. So, I mean, that you can already start doing. And even, in fact, your biomarkers, you can actually start advertising and charging people in a way for a test which is no problems with the FDA, because it's something that's done routinely every day. So I think you can start to take the steps, get a doctor involved and a researcher, set up a clinic or work with a clinic and start funding it. I don't see anything stopping you.
Okay. Well, actually, nothing's stopping us. We are, in fact, we have a core group of individuals that is doing that right now, much of that. You know, we evaluate different therapies and do biomarkers. And if anyone wants to hear more about that, please contact me privately. I don't want to throw everything we're doing out here on, you know, on this recorded. I think we're still recording. But just, you know, I'll be glad to talk about it with anyone. But if you want to be a part of it, you know, plan on bringing something to the party. It could be expertise. It could be therapies that you've developed. It could be money. So let's talk. But
well, I think, for example, you're just getting people in a consultancy with the aging, the biomarkers. You can start charging for that. Charge $10 for a consultation or something like that, and then also brand it. Call it a GRG clinic. You know, I think that's all that's really lacking, is to call it something, and then actually franchise or market that. Because, like I said, in the ending, the lifespan conference, there was this Dr. Grisson, I think, who was saying that all kinds of venture capitalists are trying to get him to franchise his wellness clinic. And he was doing exactly this: biomarkers, supplements, looking at parabiosis as a new possibility. So he was doing the stuff that GRG
What's
is
his
doing.
name,
But
Miles?
man, I have to maybe just quickly check and I'll put it in the chat.
Okay.
But I'm saying basically he
later,
was doing the same thing.
please remember. Yeah.
Yeah, I'll do that
Go
in
ahead.
the chat now. Yeah, but he was doing basically what GRG is doing, but now is a medical doctor. So you need a medical doctor to be able to prescribe a few things. But if you put all of that together, I think it's a very viable thing to do to get GRG clinics going.
Well, great. Yeah. Now you impressed me, Miles, because not only do you have great ideas, you are you do things. You're the reason we're having this meeting today. Now, would you believe I'm really, and I'll throw in another gratuitous, really busy these days. So I would look to you or anyone to maybe send me a few points, in writing as to not only what we would do, but how we would do it. But please understand, we're doing a lot of that now. And
how much funding are you talking about, Johnny?
Well, I would look to the person who is proposing the project, but how much will it cost? What, to reset the epigenome to a more youthful state? I don't know.
I don't think anybody's going to give you a quote on that. They will tell you that they want to do something in that direction to make progress, and you'll have to decide whether you want to pay for it or not. I think that's more or less the way this kind of research works.
Okay. So anyway, having said that, thanks, Miles.
I'll check with that because I've got doctors who, even in South Africa, who are able to at least prescribe the stuff. I've been to the pharmacies and they've got the supplements available. I've been to the blood clinics here in South Africa who do the same kind of biomarkers, so these things seem to be available if you just put them together into some sort of program. But I'll check. I'll give you the name now of this doctor who's doing it already. I think, let's just finish this point and we continue with the meeting.
Sure. Well, I'm
going to stop sharing now. I think we finished with this point, right?
Yeah, and I'd like to, if we're done with this point, and I mean, does anything else have any optimism in the way of gene editing, gene therapy, and my particular favorite, which sometimes disagreed with, but knock-ins of youth-enhancing genes, and I'm keeping a list of genes associated with youth from a number of sources, and I'm keeping a list of researchers active in this. But does anyone have any thoughts, positive or negative, of knocking in genes that are associated with youth, or genes that I'll say it more directly, that result in youthful expression or the expression of youthful features in our bodies?
If you could do it for 5% of the cells, would that be sufficient, or do you have to do it for every cell in the body? 5%?
Well, I don't know. We don't know till we try.
Yeah, I think at the 5% level, there are probably ways of doing it. At the level of doing every cell in the body, I don't think there's a chance.
Right. Of course, blood's a little easier to get to.
Yes.
Okay, good.
But blood is recycled every 6 months or something, and so changing blood cells is a temporary thing.
Yeah, right. But who knows? Would that 5% be enough to cause the youthful expression? And
I think it depends on the mechanism you're targeting. In some cases, it probably is. In other cases, it probably isn't. If you just need to get some protein in the blood that isn't there, having 5% of the cells produce it might be enough. But if you need to make some kind of fundamental change in the way cells work, you've got to get to every cell. And
well,
so,
you know, number one, we don't know till we try. The other number one, it's a matter of priorities. With all these opportunities, with me personally, probably with each and every one of you, we're constantly evaluating different, I'll just put it, you know, different ways we can invest our time and money, and what's the most important thing we can be doing right
I agree with
now
you about
for greater health span and greater lifespan.
I
John?
agree with you about not knowing until you try, but as a physicist, I am well aware that you have to think very hard about experiments before you do them, or else you're going to end up wasting your time.
Oh, yeah.
And so I think you need to use logic and try to understand mechanisms behind what you're trying to do as accurately as possible before you undertake any kind of mass trial. That understanding is a thing that can get you a long distance in designing experiments. So.
Wow. I'm glad you're
Johnny,
on our team.
Johnny, it's Terry, Dr. Terry Grossman.
Oh, yeah.
Terry Grossman,
He's
MD. He has a clinic
very
already.
innovative.
He has a clinic already, and he speaks about the same things that GRG speaks about. He's doing the same stuff. Being a medical doctor, obviously can go a little bit further, but I don't see why we can't follow him. He was saying he's resisting lots of venture capitalists who are telling him he must franchise his clinic. So I think, yeah, I mean, he's not taking chances. He is. If you go through the conference, he explains why he's been careful, because he can lose his medical license. But despite all the issues, he's been having the clinic in the US, in Colorado, and he's an anti-aging specialist. He says that it's an anti-aging clinic.
Well,
So
actually, several years ago, I met him at a conference and signed him up for our GRG list. I doubt that he ever actually reads. Well, maybe he does. You know, there are people, you know, who read our messages, and maybe once every year, once every 5 years, they'll pop up with either a message to GRG or a little note to me. So, but yeah, thank you for reminding me about the innovative work he's doing. Also, yeah, Eric Verdon, he's outstanding, doing some innovative things. And there's another guy I want to tell you about, although he was impressive, very expensive. I'll bring his name back to mind in a few minutes, but he's worth learning about. So, okay, well, let me share my screen for a moment, because this is the thing I said I would show you earlier. This has to do with the proteins. Just a little backdrop. A outstanding scientist, Benoit Lahalier, who is now working for Alk. And what it does is it returns the values of over 4,500, something in the neighborhood of 4,900 different proteins. And let me show you a raw file. This is actually the output. I think it's an Illumina output. I guess that's a standard called ADAT, but there's a way to convert it to an Excel spreadsheet, and it's just really difficult to decipher looking at the raw ADAT file. But basically, this is what it looks like. And what do we have here? It's a bunch of blank space. Well, we go over here, and here's the actual data. And some of these targets, you protein gurus, these may look familiar, but CRBB2, CRAF, ZNF41, I don't know, but these are different proteins, and these are found in the blood that this SomaScan returns. Now, what Lahalier and Weiss-Corey and others was brilliant. They identified, I think the number is 490, that are associated with age. And this is the list. Now, the one CD38 that I think it was John talked
Yes.
about, is there another name for it? Because I've searched for it here, and let me try and find it in the C section. Oh, wait, here we are, CD36, CD58. So they didn't use it. Hang on, let me go back to this. Okay, it is returned by the SomaLogic SomaScan, and if we go out, there's like 4,800 of these. But, and you'll see why this is so. This is not included in the list that Lahalier list identified as the 490 ones that are, that have values that are most closely associated with aging. And I was very surprised that GDF11 is not on here as well. GDF15, I think, is. And this will make a little more sense when I show you what I'm going to show you in a moment. Yeah, GDF15. But so what I did is I wanted to, and Benoit Lahalier, to the extent of his limited time, was very generous with his time to help me to bring the calculations from the paper that they, I have. I'll show you the paper later, but this is here and now, and it's something we can do with it that relates to this discussion and something we can do to move forward. But to extract these proteins, and I was in databases, data analytics, software development, computer programming for 20 years while setting the stage to just quit that and solve aging. But it took to extract all this, because you have to develop a process to extract this, to get it into this format. But the SomaLogic rep provided me with this file, which I don't know where this data came from, but what it is, it's, wait for it. It lists the these are the subjects here. It's listing the chronological ages. And then, okay, this subject, 59-year-old male. Out here is the actual data for the proteins that we saw for all these proteins. Okay, don't try and wrap your head around that. It'll set you spinning.
They're using some kind of biochip to do the measure the proteins in the blood for all these subjects. Is that what's going on?
Yeah, I think I don't want to misquote. I think it's the Illumina chip scanner. I don't know. I don't remember. We can look it up. But that's the important thing is that I extracted very tedious. Oh, Helga's back. Okay. So I took this file, and it was very tedious, one at a time. I'm looking to automate this process, but I extracted 64 different of these subjects because I wanted to see with my own eyes from the SomaLogic sample file how closely this result using the Lahalier calculations could be turned into something usable. That's kind of the end of the story. So for all these proteins, here is this subject is a 55-year-old male. This subject is a 77-year-old male, and so on. So for each of these proteins, these are the values. Now there are coefficients or multipliers where you take this times this and you get a number, okay? Then you add them up. So this is the result for this person, age 55, 17,102. It's just a number.
The value is what they measure, and the coefficient is some kind of weighting factor that they came up with for some reason.
Right. It's in the paper.
Where do
it explains it.
Where do the coefficients come from?
Well, I don't recall. We can read the paper but these are the numbers that are applied to each of these.
It must have been some
multiplied.
kind of multiple regression where they were
Yeah.
trying to decide which proteins are most important for some particular thing they were focusing on. And the question is, what were they focusing on?
Well, okay. I can't answer that right now. I was more interested in recreating the process and testing, gaining enough of an understanding, recognizing there's really smart guys like you who could, evaluate, the underlying premises and so forth. But I just wanted to duplicate his process and test it for myself to the best extent possible with the tools available, whether it worked. And I'm going to get there in just a moment. So here's more of these end results. So what I did is I took these numbers and I plotted them versus age, and here's the result. And I believe I reported this to GRG previously. I probably did not, get with all the messages, I did not give in-depth, description of what I was doing, but I just wanted to cut right to the end result. And here it is. So lo and behold, there was roughly a linear relationship between these 64 different values and a chart. Now, it's kind of like this in real life with DNA methylation. You have some outliers. For example, this 68-year-old who, the lower the number, the better. And this 71-year-old, they're probably doing some things right. Oops. Okay, I'll repeat that for Rolf. Hi, Rolf. These two older people, the lower the number, the better, according to this model. So these guys are probably doing something right. Now this 45-year-old is doing some wrong things, probably, or maybe has a disease condition. This person, I suspect, this 44-year-old, his number is over here somewhere with the 70-somethings. I suspect this is, if he isn't now, he's going to be one sick puppy someday. And he should either get medical treatment for a disease or probably several comorbidities, or get on a serious, nutritional exercise program, and possibly when the fundamentals, are, stress reduction, whatever, are brought under control, then maybe some other things like, we do in our programs. But notice this 18 and this 22-year-old, how low their numbers are. They're in the negative. Now,
that's where we want to be.
Well,
Yeah.
I guess so. Now, I'm going to throw in a caveat. All this, I start thinking, okay, so what would we do to turn this into something we could all use to stay younger longer and increase our function and joy of living and all the end results we're seeking here, in addition to intellectual stimulation and companionship and all that good stuff. So hence the question I asked earlier about, okay, we could possibly, and this is another thing my foundation and the folks and I are working on, is how to increase values of things. For example, those of you like me who are injecting GDF11, unfortunately GDF11 isn't on here, but who knows what changes in these proteins GDF11 is triggering. Similar to Harold and Akshay's. Well, I understand it's the name has been changed from Elixir, but that, or the convoys and Dobry-Kiprov's therapeutic plasma exchange, or the umbilical cord plasma infusions that I do and others in our group sometimes do, and possibly some of you do as well. How is it going to change this? But given if we want more of something, how can we identify the most important proteins first, and then all of these proteins to have a protein cocktail that's tailored to us depending on our values to make us healthier and happier for longer. And also the other question was, how do we what's the word, capture or damp down proteins that are high that need to be lower, should be lower. So that's one question. The other question is there something about our proteome that serves a function to keep an unhealthy or an older body, an older genome, who knows what that's going on in a person when they're older, sort of in check and keep it healthy? Would, if automatically I could make make my protein profile the same as let's find the 22-year-old here. Yeah, here he is, the same as this guy. If I could reset my ACAN, agricam core protein to 999.6, my ACBD7 to 80, so on down the line, would I be healthier or would it make me sick? Possibly kill me. So these are the things I think about when, number one question in any therapy, well, one of them is it safe? So I'm going to shut up now and let the rest of you talk. What do you think of what I've said, and how are we going to use this?
I think it depends.
Okay. Go ahead, John.
These protein biomarkers you're looking at, are you sure they didn't establish those coefficients by doing an age regression in the first place? I mean, are you just reproducing what they did to generate those coefficients, or are you doing something different?
Good point. It's coming back now. I seem to recall they analyzed the proteins in a large number of subjects, and the younger ones had, well, here I'm applying the coefficient to arrive at this to each of these. You see?
Yeah, well, I was asking where the coefficients came from.
I think that they analyzed it directly, similar, to the Levine, Horvath, and now we call it Kramer phenotypic age calculation, to reverse engineer it. But what I did is I reverse engineer it on a sample set to see what happened, and this is what happened.
Okay.
But yes. I'll get you the paper. I'll send it to GRG.
Okay. Yeah, I wanted to mention a slightly different but related thing that was at this ending age-related disease conference I was talking about. There was a guy named Daniel Belsky from Columbia University who had some data about how fast the epigenome age is changing rather than what the absolute value of it is. It's sort of he was looking at sort of the slope instead of the value, the derivative, and he has come up with a way of taking the methylation data from one of these DNA methylation chips and using it in a different way than Horvath has been using it to extract the rate of change of the biological age rather than the absolute value of the biological age in a way that's independent of the biological age. And there's two interesting things about that. One of them is that it seems to be more sensitive to doing supplements and innovations than the the epigenetic age does. So when you do something good, it shows up immediately in this measure. The other thing is that he has interested a company in providing these values. But the problem with that is that it's not very cheap. The company does DNA methylation age for $399, and for an extra $75, they'll give you this. And what you'd really like to do is do this before and after some intervention to find out if it's working. But that means you're spending almost $500 a pop to get this value. But anyway, I will put the reference to the guy on the chat so people can chase him down if they're interested.
I'm interested.
Question for you, John, since I think you know the math. Would that be some sort of eigenvalue analysis of that matrix? Which proteins have the most effect on the age or on the aging? Some sort of eigenvalue analysis on the matrix?
Something they must be doing something like that. There, as you know, there's a whole toolbox of mathematics for measuring correlations between one thing or another, and that's one of them. Multiple regressions is another, and so forth. And it's a little hard to know what they're doing, but yeah, I think identifying a few proteins that would be rather than taking the whole biochip, and which is really expensive. These biochips cost thousands of dollars, and you can do several different tests of different individuals at once, but nevertheless, the price is on the order of $500 to $1,000 every time you do one of these measurements. If you could identify a few proteins that were the sort of key things that were moving in the right direction and analyze those, you could do the same kind of analysis much cheaper. And it would be nice if somebody could figure out exactly how to do this and offer it as a service for $50 or something. But I don't have any idea whether that's likely to happen or not.
My comment to Johnny is that I must say, I'm sure you'll be healthier if you revert to your 22-year-old profile, because like I've said, aging is effectively a neglect of the biology. It neglects you after a certain age. It doesn't care what happens to you, because the biology has the benefit of the half-life of the young individual.
I think I think you're programmed to die. I think it's not that it doesn't care, but that it's specifically changing your epigenetic pattern to make it harder and harder to live, and therefore one needs to be able to reset that epigenetic programming so that process stops.
Sorry, Johnny. Sorry, Johnny, for that news.
Yeah. I have a question.
Wait, Miles, what are you sorry about?
I'm saying sorry. John says you actually programmed to die. Sorry about that.
Yeah,
it's nothing personal. It's nothing
That's
personal.
how I feel about it. Hey, I had to turn on my air conditioner. Can you hear it? Is it loud? Too loud?
I don't hear it.
Okay, go ahead.
Quick question.
John? Yeah.
Yeah. John, when you say biochip, is this a biochip you put into a beaker of blood, or is it a biochip you put into your arm and you wear it the rest of your life?
It's a thing you take blood and you extract something from it and put it on this chip, and then you look at it with ultraviolet or something, and different pixels light up, and you measure how brightly they light up, and you make a matrix like Johnny was showing.
So it's basically a piece of chemical apparatus.
It's a chemical apparatus that does, or at least in the case of DNA methylation, it does 100,000 tests on one chip rather than having to do them one by one. So it's an enormous improvement in efficiency, but it's not cheap.
Yeah. And how far away are they from the implantable version? That's because that's what we need. We need real-time measurements.
I have no idea, but probably if somebody was willing to spend a lot of money developing one, it's probably possible to do it, because I would think that if you can do something that lights up under ultraviolet, you can also do something that gives you an electrical signal from a pixel. scope of a scan. It shouldn't be all that much of a different
Right.
technique.
And if that were the case, you wouldn't need to have 10,000 things measured. You might look at the top 10 or top 100 things being measured, have real-time measurement, and then you can start applying treatments and getting data very fast.
Yeah, well, it also appears that these things tell you more than one thing. I mean, you've got a lot of data and you can analyze it in a lot of different ways. And this business about measuring the slope of the change in the epigenetic age rather than measuring the age itself is an example of measuring two sort of orthogonal things that you can get from the same database of chip readings if you're clever.
Right. I no longer remember any of my physics. I only took 4/4 of it, but I recall that basically that basically the things are related. There's distance and there's velocity and there's acceleration and there's jerk, and so these various quantities are used in the aerospace industry and missile guidance problems and that sort of thing.
Yeah.
And if you have your and when you're a student of calculus, you appreciate that when you look at all of these things together, the distance, the velocity, the acceleration, and jerk, then you get a really good picture of what's going on,
Right.
much better than any one. And your point today is that they're just by going distance is the analog to just measuring the steady state, and the velocity is the rate of change. And so they've got two of the four metrics that people do when they're designing missile systems. And so we have aging is obviously a dynamic thing. I fear you're right about we're programmed to die, and it's not our job, but it's our consciousness will be a lot happier if we find a way to change that programming and overcome that programming. And so I'm just thinking, kind of riffing, if you will, about the idea here that it would be really great to get perhaps a smaller biosensor that just got some of the top candidates, but actually it may not be much harder to get a 10,000 metrics or things measured than it is to do 10 or 100, because we're talking about microelectronics.
Yes.
And it may be equally hard to do a few as it is a huge number.
Yeah,
Yeah,
the way
I
big
think
problem.
that's the principle that Soma Logic incorporated. Like with their Soma scan, you get like 4,900 different proteins. Oh, and in terms of actually taking this the next step further, I've had discussions, you might say negotiations with their reps and them. I've gotten the price down to $850 per test with a minimum of 15. Plus there's a certain amount of blood processing. So it takes a phlebotomist that this is standard stuff, but they have to spin it with a particular kind of centrifuge. And
Are you sure they can't do saliva?
I'm sure. Well, they probably can, but their process that we believe works. But
If you're going to market it, do mass marketing, I think the phlebotomy is a barrier. And if you could do something even a little worse with saliva, it would be a real advantage.
I agree.
Well, absolutely. Oh, heck yeah. But so I've had that waiting in the wings for a couple months, and amid all these different projects that I have going on and different priorities. But so 850 times 15, but I've had experience before with these companies that it looks great. They're doing really innovative, cutting edge stuff, but the variability is all over the board. So yeah. I would want to, of the 15, to at least 3 of the subjects to be duplicates. Probably better to have 4. So there's like, a duplicate. So that's kind of like 850 times 3 or 4.
Even better, do it with another company.
Even do what?
Even better, do it with a second company.
Well, there is no, I don't know. I've been looking for one. Believe me, if you can find one that will analyze those proteins, or at least the 490 that LaHallier says are important. I've been looking, please find one for me.
But maybe everybody else is. And I had my DNA methylation epigenetic age measured by 2 commercial companies. And one of them said I was 26 years younger than my age, and the other said I was 5 years older. So
Well,
I,
yeah. These methods are all over the board.
Oh,
I've gotten
I think these
different.
people are using an 8-ball or something to do their measurements and giving you whatever result turns up, but I don't know.
Yeah. Well, yeah, and I've had, a number of people that I've facilitated their, getting their DNA methylation age, kind of had to break some bad news. They thought they were doing all the right things, but you're almost, your methylation age is almost your cron age, or maybe you're a little older, but the way I couch that is, okay, that's just a starting point. How do you feel? Is your quality of life, good right now? That's great. Let's do what we need to do to maintain that. The important thing, the important thing, everyone, is you have a number, and it's one of a number of numbers in an aging, in a lab report, in an age management panel of tests, some lab work, some other functional. So let's work on shifting that towards youth. Let's work on that DNA methylation number, getting it younger, and then these other functional numbers, let's shift them towards youth as well. So if you ever have to explain to someone, that gets an undesirable number, I'll suggest you couch it in words to that effect. So anyway, I kind of went off on a sidebar there. Where were we? Miles, did you have something? Yes, no?
No, I've got nothing. Thank you, Johnny.
Okay. Well, I'm ready to move on to the next topic, but does anyone have anything else in this topic?
Yeah, Johnny, what I would say is we have to think about whether we are really doing research, which is basic research, understanding what biomarkers correlate with age. And that is where academics do. Or are we looking for the things which are at the cusp of, are we ready to use it practically? And those are two very different questions. It looks to me like this question is a research question. It's not quite ready at the cusp of translation yet, but we can definitely facilitate that. And then going to therapeutics is a whole different mountain. Once we understand, even validate what the changes are, to then make the next research question of, if you change them, does that change aging markers? Is it different question? So I think we are early days is what I would say.
Yeah, well, the first question you mentioned, the research question, is that the Lahalier protein analysis I showed?
Yes.
Well, that is ready to be taken to the next level in practical application. And the next thing we would do is we would gather 15 minus 3, maybe minus 4, just to be a little safe, 11 subjects, and actually do this. And 4 of them, just so we're kind of a cross-check, to validate that the numbers come back, we would send blind samples for those 4 subjects to verify that these values that come back are within, a reasonable variation. So, I've been ready to do that, but 855 times 15, so what's that, $12,000–13,000 plus the time and the management that it takes to do that, the opportunity cost of putting other projects on the back burner. But if any of you is really gung-ho on doing that, send me an email or call me privately. Let's talk. This is, it's really important, among the other really important things that
yes,
we're doing.
I would think it would be a good idea to do the DNA methylation age at the same time, so that you
Right.
have two values using 2 different methods.
Yeah.
yes.
Well, yeah.
Cross-validate. Yes, that would be good to cross-validate using different techniques and correlate and see how they compare. Thank you.
Yeah. Okay. To first answer part of Mike's question and then lead us into the next segment, I'm going to share my screen. This is like my foundation's website. if you're ever wondering what the heck Johnny's doing, you can click here. And this will talk about, recently I've been self-experimenting along with a lot of others on GDF11. And this talks about very briefly my results. I'll show you the test data if you're ever interested, but also I'm on my 7th month of peptide therapy. See how that goes. Also senolytics, dasatinib plus quercetin and umbilical cord plasma. But, you know, these are a couple of things I've written that facilitate, and it's a bunch of other stuff. So going back to get at a section that I believe, pretty sure is going to answer in part Mike's question, aging intervention therapy system or program system therapies metrics. This is the here and now of what I do. And, you know, friendly disclaimer, this is not medical device, this advice. It's what I use in my own age management program. It's for your consideration. Pay very close attention to safety and having a physician oversee you before you try to do any of the things I'm doing. Oh, this is an open invitation to scientists who are aging solution creators. I and my core group of individuals and all of GRG can quite possibly facilitate your work. Just have something that's long and the short of it that's going to make us young again. You know, we don't have time to mess around with a lot of academic, early stage, you know, worm research and that sort of thing. And also any others active in aging, just let's partner together. What are you doing? Let's talk. But mission, end results we seek. Yeah, I'm gonna talk about this a little bit. In the midst of all these, you know, all these tests and all the thinking, you know, it's important to stay focused on the end result we see. I break them down to physical things like mobility, senses. We want to keep our vision, our hearing, healing, you know, rapid recovery, you know, freedom from infection, pain-free living, then cognitive, mental function, clarity of thought, then emotional, and even what, you know, what you might call spiritual. And here I talk about those. You can read this anytime. This is updated frequently. Core concepts, safety, self-reliance, and hey, let's face it, aging is 100% fatal, but you know, anything we do involves some risk. We get into a car to go to see our age management physician. We're taking a calculated risk. So there's that. And this goes on to part various background. Then the second part is different therapies. I'm going to skip that. Part 3 is what I've done over the last 20 plus years. But part 4 is the metrics section. And I'll just hit a couple of high points. The reason for this is you can't manage what you don't measure. And so we measure lab markers like DNA methylation that we believe are associated with age. We believe if we can bring that DNA methylation age to a lower number, that we will have a longer, healthier, happier life. But recently, I've been focusing more on functional tests of cognition and physical functionality. But now, here's something that's useful, and like Dan Belsky's, this relates to a couple of things that we've heard today, like Dan Belsky's. When I learn of a new biomarker or objective measure, here's some questions I ask. And if it's a test, what's the variability? Are we going to have to take 2 or even 3 of them and average the results before and after? How much that? What's the cost? Does the test? You know, a lot of other. You can look this up yourself. This is available. It's my gift to the world. and other questions that I ask. So here are different categories of tests that I use in my foundation's research, as well as my own personal age management program. You can read this on your own. Most of you probably do, most if not all of these, but first, test for safety. We want to monitor liver, kidney function, other kinds of function to first do no harm. We can usually get those in a chemistry panel and complete blood count, which is pretty low cost. And then here are some other types of tests.
You went silent.
Oh, can you hear me now? Can you hear me now? Yes, no, anyone? Can you hear me now?
Yes.
Okay. If anyone can't hear me, give a shout. I don't have a chat window open any longer. Let me see if I can find that.
You went away and then you came back.
Okay. Well, I'm back. Okay.
You were talking about inflammation when you went away. Is that C-reactive protein or is it something else that you're measuring?
Oh, well, C-reactive protein is one. It's my favorite measure of inflammation, but there's others. TNF-alpha, fibrinogen, IL-6, the other ILs, and then that Myriad RBM has a test for.
I might say that generally, if you have a high CRP, then what you should do is break it down into some of the others, because the CRP is a sort of conglomerate measure. So you could do there's a subcategory of 3 or 4. LEF describes that and suggests that. I've done that 1 or 2 times.
Oh yeah, if you have high CRP, like over, in my estimation, over 1.5, but for me, over 1 for some reason, yeah, I would look into it. So
it can go really high, and I note that in the Levine measure that they take the logarithm of it in order to keep it from getting completely dominating everything else.
Yeah, well, Levine Kramer to give everyone, Levine Horvath Kramer to give everyone credit.
Whatever,
but I'm getting to that.
I did the following experiment with that spreadsheet. Was I changed each of the values by 5% and looked at who had the biggest effect on age. Interestingly enough, one of the things you have to put into the spreadsheet is age, and that has the biggest effect on the output age. But the 2 things below that have the biggest effect are basically based on blood geometry, the size of the white cells and the variation in size of the red cells. And I asked Horvath why those were the dominant factors, and he said he didn't have the faintest idea. That's just what came out of the computer.
Yeah.
But I think it's interesting that the things that most affect your Levine age are the geometry of your blood, which I haven't ever seen anybody discuss such things in a publication.
All right. Well, we're going to get to that calculation pretty quick here. And I'm gonna finish up on this because all of you can read this on your own. I've found, let's face it, we get Zoom fatigue, we get nutritional supplement fatigue. I do, taking all these pills all day long. We get test fatigue. So we want sometimes, just for particularly like a nutraceutical or something we think will have a quick effect. Here's what I do as a simple yet effective test set for some therapies. First, I start off with mental, this trail making bee, and here's a link to where you can download it. It's a time test that's kind of like finding your way out of a maze, I guess. The other one is this human benchmark tests that are online, and a physical exercise stamina test, which I do it using an elliptical machine. And basically I do 3 high intensity bursts, and I count how many strides I can do. I mean, this is all out. how many strides I can do. And there are things that I've done that have had a, you might not call it significant, but an athlete would call it significant effects on that, but also grip strength and the time you can stand on one leg. That's pretty telling for balance and strength and also heart rate variability. But here's the larger list of biomarkers that, in our small studies in my foundation, we've used. And this kind of starts to touch on Miles's, oh, there you are. Miles's, what Miles has mentioned before, that, yeah, we're doing a lot of this, but so this is the list of a larger set of panels, and everything's out here, chapter and verse. If there's something you don't understand, just contact me, and I'll be happy to help. So I believe that answers Mike's question about, at least some ideas for a test set for biomarkers. Any questions about that before we move on? Go ahead.
That's great. Thank you again for making it available. It's a great resource. Thank you.
Yeah. Okay, let's get rid of this. Let's move on to aging metrics. The concept behind this, if you go to agingmetrics.org, you'll see what it is loud and clear, is that our associates, the members of donors to the foundation, GRG members, and I want to help facilitate not just research and development, but the creation of solutions sooner rather than later that we and everyone in the community and that the world can use for greater health and longer, healthier life. So this pretty well breaks it down. There are two categories. One is biomarkers and objective measures. Now, I have relationships with a number of biomarker companies. I am what I call a facilitator. I don't like the word independent representative because I'm not in it for the money. I'm solely in it to facilitate the work of scientists. But for lack of a better word, it's an independent distributor relationship with a couple of companies with very innovative biomarker systems. And I have relationships and I make introductions to others. I'll say though that I'm very impressed with what the Clock Foundation is doing. And if I were to choose only one DNA methylation company, I'd go with them. And if you want me to introduce you to Bobby Brook or the other folks there, I'd be happy to. And if your research, they're particularly interested in partnerships with companies that are doing the kinds of innovative research that I'm interested in partnering with and helping. So I would like to introduce you. And also Steve Horvath is very much in a leadership role there. And if appropriate, I would introduce you to him. A lot of you already are working with them. So, that's great for you. So biomarkers and objective measures. And I'm here to help with that. The other, data management and analysis and other software systems. And I'm going to show you some things that I've created. I dusted off my 20 years of programming and data skills to do some kind of neat things, which some of it's available to anyone right now. Others I would do custom work for, depending on, the value of what you're working on. And also, all of this is at low or no cost and possible funding provided by the members of donors of our foundation. So these are examples of the aging therapies we're particularly interested in, the kinds of things we've been talking about today. I don't remember how much of this I've shown before. This has been refined somewhat, so But for those of you who didn't see this before, here's a little more of a list of, yeah, it's basically the same list you saw. This is a summary page of the protein clock, or the protein age analysis system. Oh, here's the paper. So John, you can just go there to pick up the paper. And if
Okay.
that's not enough, I have a couple of other resources, but this is where this was drawn upon. Calculate phenotypic age. This is available for anyone. And I think thanks to Pete Cooperrider, who didn't have an uploadable file, but he entered the data in here and it calculated the Levine-Horvath-Kramer phenotypic age. And what I did with this, let's close. No, we may need that later. Yeah, here is my most recent life extension age management panel. You can do this with just the lower cost CBC metabolic panel from life extension plus the C-reactive protein. If you don't have a current CRP, you can, game the system by plugging in what you think yours is, or a more recent one, but it's much, much better to have recent data. But this is my lab report. So what this does, it's kind of cool, if I do say so myself. We can choose this very file. And this is what I received from, when I ordered it from Life Extension. This is the lab report. And it loads here. First, notice all of these are blank. We're going to go in and extract in text format. We're going to extract the data from this file. Well, trying to get it to scroll. So that little bit of data magic. And there we have it. This is the entire report. And here it extracted, for example, my C-reactive protein, 0.56. Hey, I'm kind of proud of that. These are the blood markers that John was talking about. Mean cell volume. I think he meant this one, 96.
Those two.
RDW, red cell
Those are the two that have the biggest effect on the results.
Yeah. So those are the numbers. Here's my chronological age. How the hell did that happen? But anyway, okay, so now we're going to calculate the endpoint, phenotypic age, and the difference between the two. Bang. So according to this calculation, based upon, which they extrapolated from these whatever thousands of subjects, my calculated phenotypic age is 57.35, and I am 14.28 years younger than my than that chronological age. Thank you very much. So, and interesting is I'm going to show you, this has been slightly declining, not the precipitous drop that I'm shooting for, that I really want, but there's a glimmer of hope with the things I've been doing lately to hopefully make this happen. Hopefully it's not all by chance. So
have you measured your methylation age, Johnny?
I'm gonna real soon. Oh, well, actually I have about 3 months ago, and it is not this. It's only like a few years younger. So what's up with that? That's why what I do is I get just a number of measures, these, lab type measures, this calculation plus functional tests, and form a composite. And do them before a new therapy, then do the therapy, then at the appropriate point in time after the therapy, retest. And I look for shifts towards youth in most of them, if not all of them. So let's see what happens. I've done this before on the spreadsheets. Let's see if I put 61 in here. Pretty sure we can do this. I've never actually done this. And let's calculate. It will be younger. I've done this before, but not 14 years. It'll be something like between 11 and 13. Yeah, there we are. So we're still younger. You probably did this, John, but
Yeah.
the width.
I think they're cheating by actually asking you what your age is and then telling you what your age is. But if I were doing the regression, I would have left that factor out, and I don't understand why they have it in. But anyway,
Well, hell, yeah, we can put in any number here that we want. Let's put in, just for the hell of it, let's put 11. What happens? Great. This is 6.83 years before I was born. So what's up with that? Again, the important thing is shifts in this number after a therapy. And then hopefully, we feel great. We're engaged in life. We're loving life. We're our minds are sharp. We can go where we want to go, walk where we want to go, run, that sort of thing. Go ahead.
Just for fun, put it in older age. You went to 61, 62 or whatever. Why don't you go to 80?
83.
83.
83.
Put my age.
83, all right. Okay, so the gap older is widening. So I think what that's telling me that all this becomes increasingly important as we grow older. That's what I take from this. But anyway, all you have to do is go to agingmetrics.org and click on Calc Thin Age, and you can plug in your numbers or you can upload your report. It has to be in this format. The LabCorp report you get from LabCorp won't work. I have some folks who are working with me to help me roll out and add all of these measures to the extraction and then saving it to a database. That's for something else that we're doing. But for now, this is up here available for you to use. Moving right along, here is a subject management, and this is Marcus Welby, the famous TV doctor. These are his all-star scientist cast of patients. These are all hypothetical, made up, dummy data, as we call it. Although for my own use, I did import my own data and I'm tracking it with this system. But let's take a look at Christian Barnard. And this page needs to be expanded to include some other information, but that's not important here. But let's go to what I call a timeline, because we uploaded some data from him, and he did some therapies, and he his physician, Dr. Welby, made some notes, and this is a timeline. And so this is his data. It shifted from 15, phenotypic age shifted from 59.95 to 59.09. So Dr. Welby has him on a good program that's at least moving in the right direction. I think he's probably, maybe here's his age up here. He may be keeping up with his chronological age. Then we go down here. The screen is, yeah, I can make this smaller, but this shows other data. And in this system, there are measurements in this column, therapies. He began aerobics training and he needed, in the notes, Dr. Welby says he needed 2 aspirin and a Band-Aid. I thought that was for the guy with the head transplant, but no, the full body transplant. Okay, then another measurement, and then okay, the subject is beginning a demanding 2-week travel and work schedule. I guess Dr. Barnard's going around replacing a lot of hearts throughout the world. So he began this there. So anyway, okay, enough of that. Let's go back to, there's a page here where they can add things. If there's a therapy, they'll want to add the date, the comments, and the therapy. That's pretty simple. Measurements, and I'm expanding this to a lot more things. Then finally, the notes. That's the yellow thing that we saw. Then there's another thing, charts that I didn't put here yet, but that'll be pretty cool. So my point is, these are the kinds of things that I would create, and I now have a couple of other software developers who will work under my direction. And I could probably, either I or my foundation, meaning the donors to my foundation, could foot the cost to them. So if any of you, like Akshay, are you still here? I'd be happy to help you guys with either this, implement this with a special site for you, or develop something that will help you, be more efficient and effective. And anyone else working on a truly innovative therapy. Go ahead. Someone had something?
No, I said I'm very much here, and we will follow up on that later.
Yeah. One more thing I want to show you. This is the age management telehealth physicians concept I talked to you about. I'm going to log in first. Oops. And this is my live data. I'll just show you the timeline first. Okay, that's wonderful. But here's the charts, which is, kind of a powerful way to see what's going on. And this shows, come on, the site is rather slow today. Well, this shows changes in CRP. I kind of like that drop from 1.2 to under 0.6. Phenotypic age, come on, you can do it. Don't I have that data in here? I don't know why it's not charting that. Dumb. Demos. Things always work for you, but never for demos. Yeah, so that's my albumin. Okay, it's rising. As John will attest, higher albumin is associated with, with younger phenotype, or at least younger phenotypic age calculation.
Negative coefficient, yes.
Yeah, and basically we are learning more about the functional effects of albumin. It's a good thing. It sucks up pro-aging factors in the blood. I mean, sucks up, that's a highly scientific term, what binds with and facilitates the excretion of pro-aging factors in blood. Maybe, I don't know what they are, misfolded proteins or whatever, but zombie cells, old senolytic, senilized cells, whatever the proper word is. And well, my phenotypic age is, there it is. It finally decided to load. But the phenotypic age has been declining over the last month. I want it like down here, but it's moving in the right direction. So at least there's a glimmer of hope that keeps me going. So let's see if there's anything else here that would be useful. Yeah, that's pretty much it. I'm going to stop sharing my screen now unless there's something someone wants to see. Nah, okay. So I believe, Miles, does that close up the third section of the agenda? Or is there something you wanted to move on to there?
No, I think we finished, Johnny. Now it's just general. Thank you.
Okay, John, you have a question?
Comment on. You didn't mention devices that monitor your body functions as a biomarker, but I think they are. There was a talk at this meeting I was talking about by a guy from Colombia. I'll put his information on the chat. Who has been, for a large number of people, has been monitoring their various things that you can get by a smart watch. I guess an Apple Watch is what he was targeting on. And he noticed a very interesting thing that some of the people he was monitoring caught the coronavirus, and that 5 days before there were any symptoms at all or any indication that they had gotten the coronavirus, their resting heart rate went up by about 7 beats per minute and stayed there. And this was taken as an indication that this is a biomarker showing the presence of infection. Also, people who got other kinds of virus diseases like the flu or something showed similar response. And so this is something that was alluded to earlier, that it would one of the directions in which things are moving is monitoring technologies that will tell us a lot more about what's going on in our bodies and what changes occur when you do certain things. And I think that's an important area to keep an eye on.
You know how much,
Yes.
how many people that were, I mean, I guess the question is, how big is this data set? Was it 10 people, 100, 1,000?
A couple of thousand people.
It's very impressive. That's very exciting.
Yeah, Michael Snyder. Yeah, he spoke just last. Yeah, he's doing some great things. And I've been following devices and monitoring devices, and the ultimate we're going to want is real-time DNA methylation, or
Yeah.
things like that. But I use an Oura ring to the same effect, and I'm not wearing it right now. I left it in the other room, but I do want to show you some of my numbers, because it's in many ways it's the same thing. It analyzes things mostly while you're sleeping. I've not yet found a way to effectively understand how it can be used, if it can, while you're awake. But anyway, it has two things: a readiness score and a sleep score. And I'm going to crow about this a little bit, because this morning when I woke up, it tells me my readiness score is 93. You get a little crown if it's optimal. So that was pretty wonderful. I felt, pardon me?
93 out of 100, or what's the maximum?
I think 100 is the max you could possibly be. And this is a compilation of resting heart rate while you're sleeping, plus heart rate variability, and body temperature and respiratory rate. Okay, that's one of the two that I look at every day, and this saves it for you. But also a sleep score tells me 92, which was optimal. I didn't even take any melatonin last night. And it calculates that by total sleep time, time in bed, sleep efficiency, and resting heart rate. But there is a way that resting heart rate. They are, and you can look this up if you're interested. And there are ways to monitor early, coronavirus. So that's another way to do it. And I find the Oura ring, it's comfortable. I wear it all day long now, usually. It gets a little annoying. I wash my hands a lot during the day, so it gets a little annoying with that. But I tried the Whoop strap recently, which may have been the one that Snyder uses, but the band is really scratchy, and I don't see how athletes who are sweating could put up with that. So I sent it back. So do you know of any other devices like that that would be,
I think a lot of people make watches. I guess Garmin does, Samsung does. I have a Samsung watch that measures your heartbeat in one thing or another. But I think this is a developing field, and some of them claim to measure your blood pressure, but there's some question about whether they're actually being able to measure your blood pressure or not. When I bought my Samsung watch, the advanced publicity said it would measure your blood pressure. And when it arrived, they took it back and said, no, we're not going to measure your blood pressure after all.
So please tell me again, John, what it does measure.
Oh, it's a Samsung Health watch. I for Galaxy Health or something like that.
Was Snyder's data only about the Apple Watch?
I think he had some people who were wearing one of those rings as well. Yes, the talks are online, but I'm not sure how easy it is to get to them. I had a press pass because my column and analog about such things, but I think I don't know whether after the fact whether they're making the talks available. But I can get to the online talks, but I'm not sure everybody can. Thanks.
So Galaxy Watch 4 Classic?
No, it has something, it's called Health or something like that. Well, Galaxy Watch Active is what it is. I'm sorry, not Health, Active. Yeah, that's it.
Very cool. And how much are they? 198? Does that include like software and everything that's needed? Okay. Very cool.